All right. Yeah, let's go ahead and get started. Thank you everyone for joining. This is our second Nucleate Translate session, a series focused on sort of exploring clinician careers and clinical careers in biotech. Um, we're very excited to have Dr. Kiran Musunuru join us today to talk about his work with genetic medicines and particularly focused on cardiovascular disease, and also to share a little bit about the arc of his career. A quick bio on our speaker today. Apologize, I think I lost it here. Dr. Musunuru is a professor of medicine at the University of Pennsylvania and a practicing cardiologist, as well as the director of the Genetic and Epigenetic Origins of Disease Program and the scientific director of the Center for Inherited Cardiovascular Disease Program at Penn. In 2021, he published the first preclinical proof of concept demonstrating base editing of PCSK9, a target when addressed that could durably reduce cholesterol in non-human primates. This work formed the basis of Verve Therapeutics, of which he is a scientific co-founder and senior scientific advisor. He's also an author of the CRISPR Generation, the story of the world's first gene-edited babies, as well as Genome Editing, a practical guide to research and clinical applications. Dr. Musunuru received his bachelor's in Biochemical Sciences from Harvard College, his MD from the Tri Institute, his PhD from Rockefeller University, and his MPH from Johns Hopkins, and last but not least, an ML from the University of Pennsylvania. So we're very excited to have him share his insights with us today, and I'll pass the floor over to him.
Great, thanks so much. It's a joy to be here. I'm really grateful for this opportunity just to talk about my career trajectory and how it led to the founding of Verve Therapeutics, which I should also mention by way of disclosure. My primary disclosure is that I'm a co-founder and advisor to Verve Therapeutics, as you already heard. In case that's relevant here, probably not. If anything, like that's the whole point of this, right?
Yeah.
To talk about how clinicians' career feeds and their experiences feed into the formation of biotechnology companies and hopefully development of therapies that can help patients. And so I'll give you one example, hopefully, that's instructive as to how it can be done. It's not the only way it can be done, of course. There are many, many ways to do this kind of thing. And I kind of accidentally fell into it, as it turns out. It wasn't intentional by any means. I never had any ambitions to be an entrepreneur, so I'm sort of an accidental entrepreneur. If I can even reasonably give myself the label of being an entrepreneur, and I apologize if you hear an ambulance in the background near a hospital, but it is what it is. And yeah, why don't I get started? I have slides to show, but before I launch into the slides and get into the more didactic portion, I just want to give you a sense of how I got to where I am, the beginning of the story. And so, as you already heard, I went to med school, I went to grad school as part of the tri-institutional MD PhD program at Cornell and Rockefeller. And during that experience, learned how to be a physician, learned how to be a scientist. But what I did then didn't necessarily have much to do with what I would do later in my career. And so that's perhaps important point number one. The training is one thing. What you actually do in your career can be something quite different. And so my PhD was in RNA biology and structural biology. I was doing things like biochemical assays and generating knock-in mice and doing X-ray crystallography. And it was a broad multidisciplinary experience and has almost nothing to do with what I do now as a cardiovascular geneticist and gene editor. There wasn't even really there was gene editing, but not in any practical sense way back when in the early 2000s when I was doing the bulk of my PhD research. So I couldn't even envision what I'd be doing now. And so you might think, well, since I was working on neurological diseases, because the proteins I was studying, the RNA binding proteins I was studying, were involved in neurologic diseases like Fragile X syndrome and paraneoplastic neurological disorders, you might think very naturally I would have gravitated towards neurology or even neurosurgery. And that's what I thought I was going to do. Then I went back to medical school and ended up finding internal medicine more attractive because I felt like one, as a clinician, could do more for patients. That's a very personal decision, as to what, fits best for you. And for me, internal medicine ended up being the best fit. So then I went to Brigham and Women's Hospital in Boston to do my internal medicine residency. And during my clinical training, didn't really give much thought to what I might do in the future for my research career. I didn't try to do research at the same time. I know some do. I think Again, it's a personal perspective, but I think that's a little bit of a disservice because when you are doing your clinical training, that is your time to learn how to be a wonderful clinician. If you don't do it then, it's certainly never going to happen. And you're basically consigning yourself to being not a great clinician if you don't take full advantage of the training experience. And so that's maybe point number two is that, whatever it is you're doing at any particular time, give it your all. Don't try to be a multitasker. I'm not sure multitasking is really possible given the physiology of the human brain. Some will make claims. I don't believe it. If I have any strength, it is that I'm a very good serial monotasker. Whatever I am focused on at any given time, and that can be for a matter of hours, it could be a matter of days, it could be a matter of weeks, whatever it is I'm focusing on, whether it's being on clinical service, whether it's writing a grant, whether it's working on a presentation, whether I'm teaching, whatever it is I'm doing, whether again spanning from an hour to possibly many weeks, depending on the intensity of the activity, that is what I focus on 100%. And then when it is done, I shift to the next thing. And I think that works both on day to day, week to week, month to month basis, but also I think works with respect to one's career. You shouldn't go in with the expectation that you're going to be doing everything at once because it's not possible, at least if you want to do it halfway reasonably well. But with the way my career has unfolded is I've had phases in my career. There was training, and there was my time as an independent investigator in the academic setting. And then there was my time at Verve Therapeutics, which you hear about momentarily, which was really a good year and a half solid experience where that was basically where all my energy was focused. And there was my coming back to the academic setting and then launching into a new phase of research activity. And that has worked very well for me. Not trying to be all things at all times, but, doing one thing very, very well at any given time, then going on to the next thing. Okay, so having said that, I went back to medical school, decided to do internal medicine residency, went to the Brigham, focused on my clinical training, did everything I could to become a very, very, very good clinician, then went to cardiology fellowship at Johns Hopkins, as you heard. And it was there that I really came upon what ultimately became the research problem with which I've been focused, really for the entirety of my career so far. Not that I'm that far into my career, but training path is so long. You don't actually have that many decades, as it turns out, to devote to one's career. And so I'm maybe, if I'm lucky, a third of my way through my independent career, closer to a third than a half. We'll see. But that's when I really decided my research problem. And, most people, I think, in my position, talking to folks like yourself, will tell some inspirational story about, oh, I had this one experience and it turned me on to, like the excitement of working on this problem or this or that, an inspirational story. And my story is actually different. It's actually one of sort of negative inspiration, if anything. Like it was a series of bad experiences that really drove me to where I am now. And so it was really this one very, very unusual month in the cath lab. Never intended to be an interventional cardiologist, but it's part of the general training to do at least coronary angiography and learn how to do it, and in the process of that, learning about coronary anatomy and physiology and so forth. And so I just had the worst. I think it was the worst luck. I don't think it was me, even though I was the common factor, that I was somehow terrible, 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 whatever. But it just everything lined up badly. I think it was a winter month, and so things were already sort of primed to be not so great, just from a psychological point of view. But it seemed like every week I was on in the cath lab, there would be like this one really catastrophic case that happened. The first case was a patient who had heart failure of unknown etiology. We were doing coronary angiography to rule out ischemic heart disease, very common indication for coronary angiography. And in the process of shooting the arteries with dye, one of the coronary arteries ruptured, which is not a good thing. It turned out he had clean coronaries. So he had no coronary disease. We caused really, really bad coronary disease. I said he, but it was actually a she. And that meant, instant ST segment elevation myocardial infarction, and not something that could be fixed with stents or balloons or anything like that. We had ruptured an artery. So that was emergency transfer for coronary artery bypass graft surgery in someone who was not healthy. She had heart failure, but obviously not the outcome you wanted. And then the next week it was actually eerily similar, except this time it was a gentleman with new onset heart failure, unknown etiology, who was doing coronary angiography to find the coronary anatomy. And we didn't do anything to his coronary arteries. They were fine. They turned out to be clean. But in the process of getting femoral access, must have boxed the femoral artery, because soon after this procedure, his leg got cold. And of course, that's a vascular emergency. And so he had to get rushed off to surgery. And then the next week, it sounds really bad, right? The next week, we had a patient who hadn't had any contact with the medical system for 30 years, and, had chest pain, had positive enzymes. So we needed to do some sort of coronary evaluation. And in the process of looking at his coronary arteries, and he, not surprisingly, did have disease. He had not been taking care of himself. But he had a lot of plaque up and down his aorta, and in the process of putting catheters, towards his coronary arteries, must have dislodged some plaque, and it unfortunately went to his brain, and he had a stroke, and had to, undergo the emergency protocol and TPA and all that stuff. And then the next week, there was a very, very sick patient who ended up in the cath lab. We were trying to do coronary angiography, and he died on the table. I don't think you could blame me for that. He was just really, really sick, and that happens.
Yeah.
So the point is, we like to think of cardiology as very advanced and we can do these wonderful things for patients. And most of the time we can, but often we can't. Sometimes it just doesn't work out. And this put me in a little bit of a tailspin in terms of, wow, what am I doing here? Like, what is the point of all this? And I came away really, really focused on trying, I'm being a little flip when I say this, but trying to keep people out of the cath lab. That was sort of my counter reaction to all of this happening. It's like, the best policy would be to prevent disease from happening in the first place so that they don't end up in the cath lab and are potentially exposed to these, devastating complications. After that time, I was involved in a lot of procedures and I didn't have any major complications after that. But that was the mentality that sort of came out of this very negative experience. And so it led me down the path of thinking, well, how can we better prevent cardiovascular disease? It's the leading cause of death worldwide. We're doing an okay job of saving people from the sequelae of the disease. They have a heart attack, they have, an ischemic stroke, and there are things we can do for them and we can save them with them. It used to be a 50% mortality rate. Now it's down, at least if you get to the hospital in time, it's not even a few percent. So we can do some very powerful things, but only after the disease has happened. We're actually not that good at preventing the disease. And a big part of that at that time, even still to some extent now, we don't really know what's driving the disease, right? Biologically. Yeah, we have these models, you've seen them with like coronary atherosclerosis and plaques forming and, all sorts of immune cells and this and that happening, but that's not what I'm talking about. I'm actually talking about coronary plaque rupture. What are the inciting factors? What actually drives the disease process of coronary atherosclerosis and ultimately atherothrombosis? And at the time, there wasn't a whole lot that was known. We knew about causal risk factors from epidemiology, and we had a good sense of, what patients could do to try to help themselves. Don't smoke, control your blood pressure or cholesterol and so forth. But fundamentally, we only had a few therapies. We had aspirin, we had statins, lipid lowering, we had blood pressure reduction, and that was it. And so that's a very strange place to be, given that, again, it's the leading cause of death. And it got me wondering, well, why don't we have better medications? Why, at that time, was the entire pharmaceutical industry really intent on making yet another antiplatelet agent to give patients after they've had a heart attack for incremental benefit, devoting billions and billions of dollars chasing that market, when there's just as big, if not bigger, a market for prevention? Like, why weren't the drug companies trying to develop new preventive therapies? And the answer was, we just didn't know enough about how to prevent disease. We didn't know enough what caused disease. And so I became really intent on trying to figure out, can we figure out what causes disease and find something that is actionable, that puts us beyond just aspirin and statins and blood pressure lowering medications? And so that was the genesis of the research problem. Now, the issue I quickly realized is that I didn't have any fresh insights to bring to the problem. It's not that very smart people hadn't been working on this for decades. They had. It's just a very, very hard problem. And so I didn't really know what to do. I sort of was thinking about, well, is there somewhere I can do some mouse modeling or this or that? And like, of course, mice don't really get coronary atherosclerosis. And I didn't really have a clue about how to go after this. And, timing is everything. And this is perhaps, like the next lesson I can impart. You don't have to be intrinsically brilliant. You just have to be well positioned when some new development comes on the scene. In my case, it was human genetics. The first genome-wide association studies were being done. And it was clear from the first 3 genome-wide association studies of coronary artery disease, which were all published in May of 2007, which is when I was just finishing up my first year of fellowship. that it became clear that it was working, that it was finding new risk factors, genetic risk factors for coronary disease. Each of these three studies identified a novel locus on chromosome 9 that no one had known anything about, totally new. It's still not totally clear how it influences disease, but it turns out to be the most common genetic risk factor in the population for coronary artery disease. It's non-coding DNA. Maybe it influences some cell cycle genes, maybe not. Still a little bit mysterious 15, 16, 17 years later. But the point was there was new biology to discover. And so I immediately made the decision, okay, this is the way forward. I need to learn human genetics and really get in on this, because this is how we're going to learn more about the biological basis of coronary atherosclerosis. And so then I did something sort of bold, which is I figured, I was at Hopkins, and the expectation when you're in fellowship is that you're going to stay, in fellowship, do some research years at the same institution. Once you've matched there, you're kind of, committed to being there for the next 3, 4, 5 years, especially if you're in an academic path. And I looked beyond Hopkins. I said, what is the best place in the world for me to actually do human genetics, learn how to do genome-wide association studies, actually be involved in genome-wide association studies on coronary disease, and even go beyond that and actually do biological follow-up of the things that I find. Because for me, it wasn't just enough to find some novel genetic risk factors and stop there. I wanted to find something actionable, and that meant diving into the biology. And so I quickly realized that the best place in the world for me was going to be in Boston, working at the Broad Institute, as well as Mass General Hospital, because that's where genome-wide association studies were starting to be done on coronary artery disease and risk factors for CAD, like blood lipid levels. And it was also the location of the Harvard Stem Cell Institute, because I quickly realized if I want to follow up on the results of human genetic studies, finding sites in the genome linked to disease, I would need a model system that had human genomes. And this was around the time when human embryonic stem cells were starting to become a viable model. It was just before human-induced pluripotent stem cells hit the scene. That was, coming very quickly after all this was happening. And so I needed those two components. And the only place where those two components were co-localized was in Boston. So I still don't know quite how I did it. I convinced the program leadership at Hopkins to let me go, take a permanent leave of absence, do the research part of my fellowship in Boston. They were not thrilled, but they did let me go. I must have been very convincing. In retrospect, I can't imagine how I was that bold to just pick up shop and, in what was a very comfortable situation, just, going to another city. I don't have that safety net anymore. I'm really banking on the research working, because if I don't, then I'm sorry, out of luck. I'm not going to have that nice lined up backup clinical position that one would at the end of fellowship at your institution, right? I was really going in without a safety net. And I was sort of naive and, perhaps unduly optimistic that somehow it's just going to all work out. It did work out, but it didn't have to be that way. Better to be lucky than good, I guess. So anyway, I went to Boston and started getting involved in these genetic studies, and we were starting to find things, and that was really exciting. And, initially it was genome-wide association studies, but then in 2009, it became exome sequencing studies, which allowed you to look more finely at coding regions of genomes and find coding variants, which in retrospect has turned out to be more powerful in terms of finding things that are actionable. And I'll get to that in a moment, exactly what I found and how we've been able to exploit it. And at the same time, I learned how to use stem cells. And, the stem cells weren't intrinsically useful without a particular technology that had not yet quite reached its prime, which was gene editing. At that time, it was zinc finger nucleases. Actually, at that time, it was really just homologous recombination. And I found that that did not work very well in human embryonic stem cells and iPSC. But it turned out that one of the pioneers of gene editing, particularly zinc finger nucleases, was also at MGH, Keith Jung. And so I reached out to him and started collaborating with him and learning how to do gene editing from him with zinc finger nucleases. And it didn't work that much better, to be honest, but at least that's how I got interested in gene editing. And then TALENs came around in 2009. So I was excited by that and started applying those to stem cells and spent the next few years, getting that up and running. And then, we're getting a little ahead of ourselves, but then in 2011, that was when I had my first independent academic position at Harvard, as an assistant professor in the stem cell and regenerative biology department. And then going forward a couple of years, CRISPR, of course, came on the scene in late 2012, early 2013, and I was in a position to jump on that in circumstances that I'll explain in just a moment. But the first goal was to find something actionable, find something new about the genetic basis of disease, and then be able to work on it. What I realized in doing all of this is that exome sequencing was going to be a more powerful approach than genome-wide association studies. Genome-wide association studies would find a lot of genetic loci, a lot of signposts in the genome, all of which contributed in small ways to the overall context of disease. It's going to be harder to find something actionable there. Whereas exome sequencing, if you could apply it in very, very unusual individuals with very, very extreme phenotypes, then you might be able to find a variant with a very strong, large effect on disease. And that might actually give you something that was actionable. So let me start up my slide set now, because I think I've given you enough background to give you a sense of how I ended up on the adventure that I've been on for the last dozen years. Let's fire this up. Okay. Hopefully you can see my slides. At least I'm assuming you are. Okay, yeah, good. Have the thumbs up. So really, again, it was about finding individuals with genetic predisposition or protection. Now, finding those with genetic predisposition was easy because that had already been done. Here's an example of one such individual. This is Avery. At the time this picture was taken about 5 years ago. She was 10 years old. And you can see that she's hooked up to a machine. She's getting apheresis because her cholesterol levels are so high. She has a condition called familial hypercholesterolemia, FH. She has the worst form of the disease, homozygous FH. She has a full deficiency of the LDL receptor. Now recall that LDL receptor sits on the surfaces of cells in the body, particularly the hepatocytes. Its role is to bring cholesterol-carrying LDL particles out of the bloodstream into cells. It's the way that cholesterol is supplied to cells in the body, but the liver plays an important role in clearing out any excess LDL cholesterol-carrying particles that are in the bloodstream. And when it's deficient, the LDL particles have nowhere to go. They stick around in the bloodstream. They build up to very high levels, and that puts you at very high risk of coronary disease. So she was recognized early. She, was getting, and still gets, as far as I know, apheresis every week. Arduous procedure to clean it out of her blood. Yet despite this very aggressive treatment, she had to undergo open heart surgery beginning at the age of 11, and she's had 3 open heart surgeries so far. So a very dramatic case. And, there's no mystery what's causing her disease. The question is, can we do something about it? Now, this newspaper article in my hometown paper, the Philly Inquirer, asked if CRISPR can fix the problem. I wouldn't be showing you this if I didn't think the answer was yes. But it turned out to be not so straightforward to figure it out for her. And that has to do with the biology of the gene that initially presented itself as the primary means by which one could develop new therapies against coronary artery disease. And it's a lipid-based. It's about reducing cholesterol. It's complementary to statin therapy. And we know that PCSK9 can protect individuals against ischemic heart disease. We know that individuals with single mutations have lower LDL, lower risk of coronary artery disease. There are people walking around with full absence of PCSK9. They have very low LDL levels, but as far as we can tell, they have no adverse consequences. So as a drug target, it's very attractive. There are a few other features that make it attractive as a drug target. It's expressed in hepatocytes secreted into the bloodstream, so you can target it either in the bloodstream with monoclonal antibodies, or you can target it in hepatocytes with siRNAs, ASOs, and then ultimately, as you'll see in a moment, gene editing. However, it's a direct antagonist of LDL receptor. It's in a yin-yang relationship. It causes LDL receptor to be degraded. Now, the rationale behind inhibiting this protein is that if you have LDL receptor and you eliminate or inhibit PCSK9, it will de-repress LDL receptor, cause its level to go up, and now clear more cholesterol out of the blood. However, if you are homozygous FH, rather than say heterozygous FH, and you have no LDL receptor, like Avery, PCSK9 inhibition won't work. Sorry, out of luck. You can give them medication, you can give it inhibitors. It just doesn't work in those patients. All right, so problem not solved for patients like Avery. Now, I was inspired by all this work, and I said, are there other PCSK9s lying around out there? And so what I did is hunted for individuals in the easiest way I possibly could, arguably the laziest way one could, which is I just looked in the literature to see if there were any unsolved cases. And I came upon a description of a family in the St. Louis area. This is Anna. She's one of the individuals in this family. She and 3 of her brothers, as it turns out, all have extremely low LDL cholesterol levels. So not high levels, low levels. And she was sort of accidentally found at a health fair where she was getting a spot cholesterol check. It was found to be very, very low. She was advised to see a lipidologist because person there didn't know what to make of it, except that it was clearly abnormal. Turns out she and her brothers also have very low triglycerides. And so we ended up calling this condition familial combined hypolipidemia. And the family had come to recognition in the year 1999, and an investigator at St. Louis, a lipidologist there, had tried to figure out the gene using the techniques that were available at the time, linkage analysis, single gene sequencing, was not able to find anything. But years later, in 2009, I was at the Broad Institute. There was the opportunity to be among the first people to use exome sequencing. And so I thought of this family. I said, all right, let me see if I can get my hands on some DNA samples. Fortunately, they were still lying around in a freezer at WashU, even though the guy who was studying them had retired. No one ever throws anything out from freezers. They're there forever. And so we're able to dig up the samples, getting sent to us at the Broad Institute. I was given the chance to do exome sequencing on just two individuals, two DNA samples. So I picked two of the four siblings, and we hit pay dirt. Better to be lucky than be good. It turns out that Anna and her three brothers all have full deficiency of the ANGPTL3 protein, ANGPTL3. And so some aspects of that. She and her three brothers had double mutations, had very low LDL triglycerides. We now know from further follow-up work that we and others have done that single mutations cause lower LDL, lower triglycerides, lower risk of coronary disease, as well as potentially lower risk of type 2 diabetes, all without any adverse consequences. It's a lot like PCSK9 that's expressed in hepatocytes, secreted in the bloodstream, so you could target it in either place. But the big difference is it acts independently of LDL receptor, totally different mechanism. And so you can reduce cholesterol without being dependent on the presence of any LDL receptor at all, which means that ANGPTL3 inhibition will be effective in patients like Avery. Okay, so now we have our two targets, depending on the severity of your disease. And so let's talk about gene editing. I've already given you some sense of how I fell into gene editing. It was more about doing disease modeling, modifying stem cells, putting in genetic variants, knocking out genes, differentiating those stem cells into, say, hepatocytes or cardiomyocytes, and then trying to understand the phenotypic effects of these genetic variants. And you all know about nuclease editing. That's what was initially reported in January 2013 in 5 different papers. I knew about the work a little bit ahead of time because I was on a grant with Feng Zhang, who was one of the developers of CRISPR gene editing as a tool to use in mammalian cells. I was also working with George Church, and so was able to get plasmids and start putting them through their paces and comparing them to the old gene editing tools, like zinc finger nucleases, like talons, and found almost immediately that they work far better than any of the previous tools. It's obvious now, no one talks about those older tools, but at the time it was an open question. Like, is this just another gene editing tool, or is it going to be better? Is it going to be worse? Turned out to be much, much, much better. And the mechanism, I don't think I probably need to tell this audience how it works. You have a protein component, a guide RNA component. They come together, form a little molecular machine, scans across double-strand DNA. The guide RNA is kind of like the GPS. It specifies what sequence should be found in the genome by virtue of its first 20 bases. That's the address. And then regardless of which version of CRISPR-Cas you're using, it could be Cas9, it could be Cas12a, it can be Cas12b, C, D, E, F, G, H, I, J. I'm not sure how far it goes up now. It doesn't have to be CRISPR. It can be zinc finger nucleases. It can be meganucleases. It can be tau. They all work in the same way, somehow or the other. They find a site in the genome. They make a double-strand break. Cell tries to repair that break. It's error-prone. You'll get insertions, deletions in a semi-random fashion, but regardless, you will disrupt the gene, and it can work very efficiently. You can get up to 100% disruption of a gene. So if you want to disrupt a gene like PCSK9, you can do it, or at least the potential was there. And in 2013, once I realized how well in our hands CRISPR was working in cells in addition, stem cells, of course, the natural thought was, well, how well would it work if I put it in an animal? And at that time, no one had done that. We ended up being the first to do it, to try to target and eliminate or inactivate a gene. And of course, the gene I was interested in as a cardiologist was PCSK9. And so we were so sure that we were going to be scooped, that everyone in the world was working on it, that we ended up publishing it in, a relatively obscure cardiology journal called Circulation Research, because they offered a very rapid pipeline to publication. Turned out that no one else published on it for more than a year after we published this. So, oh well. But this is the first paper reporting, nuclease editing within the body of a mouse to inactivate a gene. And what you can see here is in our first try, we basically hit pay dirt. We used the viral vector, put it into the liver, CRISPR-Cas9, guide RNA targeting PCSK9. it basically cleared PCSK9 from the bloodstream and the cholesterol levels fell by 40%. So in mice, it worked great. Now, in the ensuing years, as you all know, there's much more than just nuclease editing. Base editing was first reported in 2016 in standard fashion. I don't consider myself a pioneer. I'm not an engineer. I'm not developing new fancy technologies. I'm a very, very early adopter. So once base editing was published by David Liu's group, I immediately jumped on it and started asking, can I use these base editors, which can make specific single nucleotide changes at desired sites in the genome, sort of like a pencil eraser? Can I use this to turn off PCSK9 or other genes? And the answer was yes. And I won't bore you with the data, but we quickly showed that we could do this with PCSK9 in the mouse. We subsequently showed that we could do it with ANGPTL3, our second gene target in the mouse. We then showed we could do it with other gene targets involved in various metabolic diseases. We did in a variety of different mouse models, including fetal mouse models, so in utero before birth. I'm not really going to talk any more about that today, but it's something we've been working on. But it looked very, very promising. Now, at this point, I'd moved to Penn. I moved to Penn in 2016 from Harvard. This stuff with targeting things in mice started as a hobby, but it took on an increasingly important role in my research program as it looked better and better and better. And, you know, around 2016, 2017, I got to the point where I felt like I could do everything possible in the academic setting and wasn't going to be in a position to take things any further. That it was going to be, you know, too challenging to try to do it in academia. It would take too much funding. It would require the kind of work, drug development work, that academic labs are just not geared for, both because of the resources needed, but also because of the know-how. I had no experience in drug development. So I had taken it as far as I could go. I got together with some of my colleagues, and on the basis of some of these data that I've shown you, we ended up pitching different VCs, most prominently Google Venture, now known as GV. And in fact, we ended up incubating, at least on paper. We didn't do any lab activities. We were mostly just talking and discussing and planning out the company. But we incubated a GV for a while. They ended up becoming our biggest financial backer, had our Series A in early 2018, and then kicked things off. I rented some space in the incubator facility, the incubator space at Penn called the Pennovation Center. Got some dirt cheap lab space, something like $5,000 a month to get out of like a full laboratory that could fit 6 people. It's like less expensive than paying rent for an apartment for certainly for, you know, that many people. And you know, equipped out the lab with you know, some more funding. I sent a postdoc and a technician from my laboratory who knew how to do everything to become the first employees of the company. I hired a few more people locally, and then off to the running. You know, started doing lots of work. And so this was the genesis of Verve Therapeutics. The goal from the start was to protect the world from cardiovascular disease, as you see in the mission statement there. But we didn't really know how we were going to do it. So this is an important point. We knew we wanted to knock out genes like PCSK9 and ANGPTL3, but it was not clear at that point what the best approach was going to be, either with respect to editing or with respect to delivery into the liver where we wanted to go. There were, even at that time, a lot of CRISPR-Cas options. There was Cas9, there was Cas12a, there was Cas12b, and we ended up playing with all of them and basically doing bake-offs to figure out which one would work best. And then there were the base editors, cytosine base editing and adenine base editing. And then, you know, anything goes. We even looked into the possibility of using zinc finger nucleases. Whatever was going to work best was what we were going to use. We can go in with any particular prejudice. And then for delivery, we thought about AAV, but it has its disadvantages that I won't go into here. But then we explored what at the time was a relatively new technology that no one had heard of, although given the events of the ensuing years, everyone's very familiar with them now, and that's mRNA lipid nanoparticles. And so we put that through its paces.
Oh, I think we might have lost you, but
How long did you lose me? For a few
the
seconds?
audio was fine.
Okay.
I think you were just freezing on video, but I think we're back
Okay.
now.
All right. So I was talking about starting up Verve and how we were doing bake-offs. You got all that? Okay, good. So it sounds like you only missed me for a few seconds. All right. Sorry about that. Let's go back to the presentation. I'm not sure what happened there. Fortunately, I was able to see when you dropped off. That's good. Okay. I'm assuming I'm coming through clearly. You can see the slide. Thumbs up.
Yep, we can see the
Perfect.
slide.
Okay. Yeah.
Yep.
So we did these bake-offs, and what we ultimately realized is that the best approach for targeting PCSK9 in mice, and then, as I'll show you now, non-human primates, was to use lipid nanoparticles and mRNA and use base editing, specifically adenine base editing. So we started our activities, as I said, in 2018. We did our first couple of non-human primate studies in 2019. The second one in November of 2019, and it hit pay dirt. We got almost saturation editing of the liver on our second try, which was really good and got us very excited. And momentum had built up enough that we realized, okay, this is going to work. We just need to push and use our funding, hire as many people as we need, and just grow, and take this to the clinic. So ended up moving the lab from Philadelphia to Boston so that we could rapidly hire specialists in gene editing and RNA therapeutics from other companies, because Boston is Boston. So rather than trying to develop our own, just steal them from other companies. And then by late 2020, we had some very, very good data in non-human primates, and then submitted it, as you can see here, to Nature and published that in May of 2021. That's the top paper, to which I contributed. I had planned to take a sabbatical in 2020, a part-time sabbatical, but because of the pandemic and everything shutting down on the academic side, including my laboratory and everybody working remotely, I basically took a full-time sabbatical, unplanned, from the beginning of 2020 to middle of 2021. And that 1.5 years ended up just being decisive. Got everything working in monkeys, as I'll tell you momentarily, both with PCSK9 and ANGPTL3. Got wonderful data that set up IND-enabling studies that ultimately led to initiation of a clinical trial that I'll also describe in a moment. We had our IPO in June of 2021 because we had such compelling preclinical data and such a compelling story to tell. The paper came out. The paper on the bottom was from an academic group at University of Zurich. They reported something of the same thing, but being an academic group, they were in a position to do, use dozens of monkeys and use the latest, greatest cutting edge technology. So their results were much less impressive than our results, but entirely understandable since they were an academic group with limited resources. And I suppose if I had stayed on the academic side instead of co-founding Verve, I would have been trying to do the same thing somehow myself. But I was at the company, and so we're able to do things at scale, use lipid nanoparticles. On the left here is the LDL particle. That's what carries around cholesterol. That's what we want to try to reduce. And the point here is that lipid nanoparticles are actually very similar in some ways to LDL particles. They're basically a lipid shell, and it contains a messenger RNA for, in this case, an adenine base editor, and then a guide RNA. Okay, hold on a second here. Okay, so adenine base editor, guide RNA. I won't go into the specifics of exactly how it works. I'm just going to say that this allowed us to convert one base into another base, exactly one base pair out of 3 billion base pairs in the genome. In a location in the PCSK9 gene, the splice donor of exon 1, that was enough to disrupt the gene, and we're able to do it very, very efficiently. And here is one of our first non-human primate studies, a long-running study. And what you can see here is when we treated the monkeys with these LNPs, the amount of PCSK9 protein in the blood fell by 90%, so almost clearing it from the blood. And these are data out to 8 months, and Verve has shown follow-up data, even longer studies going out to 2.5, almost 3 years now. Once it is down, it is down. It truly looks like it is a permanent effect, lifelong reduction of LDL cholesterol levels. Now, this is monkeys. And that was the PCSK9 protein level. Now, let me show you what we actually care about. LDL cholesterol fell by 60%. And once it was down, it remained down. So one and done, as we like to say. That's monkeys. But then we were in a good position to launch a clinical trial, and I'll tell you about that. But first, I want to talk about our second gene, ANGPTL3, because this turned out to be much more challenging. And there is a reason for that. Now, when we took wild-type monkeys, healthy monkeys, and targeted ANGPTL3 again with base editing guide RNA, it worked great, 96% reduction. But here is the problem. Remember, I told you LNPs are very much like LDL, and it turns out the way LNPs get into hepatocytes is via the LDL receptor. And so you might predict that in patients with homozygous FH, therapy is not going to work because the LNPs are not going to get taken up by the liver. And that's a problem, right? So we had to solve that. But first, we had to confirm to our own satisfaction that this is a problem. And we needed a model system in which to study it. Now, the problem is that in order to really get a sense of what's happening in human beings, we need a primate model of homozygous FH, but it did not exist. But our innovation was to say, well, we just figured out how to knock out genes in the liver and do it with lipid nanoparticles. Why don't we use that very same approach to do the opposite? Instead of correcting something or having a therapeutic effect in a monkey, why don't we actually try to give it disease? So using just standard Cas9 nuclease mRNA and 2 guide RNAs to cut in 2 places in a gene and basically cut out a portion of gene, we targeted the LDL receptor gene to try to cause disease. And it turns out this double scissors approach of cutting in 2 places works very well. And when we gave this LNP treatment to wild-type monkeys, it basically got rid of LDL receptor, 94% reduction in the whole liver. And LDL cholesterol levels over the month following treatment climbed 6-fold. So consistent with homozygous FH. We now have our homozygous FH model. We were then able to take these monkeys and then ask, when we use our standard LNPs to target ANGPTL3, does it work? And what you can see here is it does not work. This is what we feared. And it was all predictable. But now we had confirmation. However, in parallel, anticipating this result, folks at Verve had been developing an alternative approach, and that was using the carbohydrate moiety GalNAc, which specifically binds the ASGPR receptor that's specifically expressed in hepatocytes, and it's kind of a backdoor into the liver. It has nothing to do with LDL receptor. But if you make LNPs and study it with GalNAc, the hope was it would take advantage of this other receptor and get in and allow the editing to happen. And in fact, it did. And now you see really great editing, more than 60%. And in fact, that's a key number because only about 60% of the DNA in the liver is in hepatocytes. And we know we're preferentially targeting hepatocytes. So this means we're doing saturation editing, hitting all the hepatocytes. And when we target ANGPTL3 in this way with these GalNAc LNPs, you see that ANGPTL3 protein levels now are almost cleared from the body. And there's at least 40% reduction of LDL levels in these non-human primates. So it looks great. Okay, so PCSK9, ANGPTL3, both poised to go through IND-enabling studies and then into clinical trials. And so that was the next step. And so launched a study called Heart-1. This was getting underway after I'd gone back to Penn at the end of my quote-unquote sabbatical when my academic lab opened up again. So I have not been directly myself involved in these trials. Took on a life of my own after I did my work at Verve. But they launched the Heart-1 study in early 2022. The first patient was dosed in New Zealand in July of 2022, July 4th, which means nothing in New Zealand, but means something rather more here in the United States, of course. So it's hard to forget that date. And just a few months ago, Verve announced the trial results from the first 10 patients. And it's a phase 1 trial, so it's dose escalation. You start low and then build up as that safety is demonstrated. And what you can see here, even with 10 patients, there is a dose-dependent effect. The patient who received the highest dose has experienced a 55% reduction in LDL cholesterol levels. And if you follow that one patient, so far, Verve has presented 6-month data. You see, once it is down, it is down. It is almost certainly going to be there for the rest of that patient's lifetime. And so it worked. It works in human beings. And, there's more work to be done. Obviously, you have to do the full clinical trials. You have to establish safety. There's some other caveats I won't get into, but it's looking, really encouraging. It's working in human beings. The ANGPTL3 LNP clinical trial is going to be kicking off later this year, the second half of the year, at least by Verve's guidance, if all goes as Verve has planned. And, hoping to see very similar results there. And remember, again, ANGPTL3 is important because it's the key to treating patients with homozygous FH, patients like Avery. So it worked. That's great to see. And that's kind of like the fulfillment of my journey as a physician scientist, right? Like starting with trying to understand the genetic basis of disease and then getting all the way to the point of actually having a therapy going into the clinic and actually demonstrating success in patients. Now, there's a much bigger picture here, right? It's more than just a few patients. It's more than just FH. In principle, this could be somatic editing to treat or prevent the leading cause of death worldwide. 18 million people die each year of atherosclerotic cardiovascular disease. And now we have a potential one-time therapy to help these people. And you might ask, well, where do you start? Well, of course, we're starting with patients with FH. They have a genetic condition. It makes sense to give them a genetic therapy. They have the most desperate unmet need. But then quickly after that, I think we'll be able to, or Verve will be able to go to secondary prevention. Any patient who gets admitted to a hospital with a heart attack, yeah, they get their balloon angioplasty, they get their stents placed in their coronary arteries. That's all great. But that doesn't prevent future heart attack. Right now, when we discharge one of these patients from the hospital, it is standard of care to give them a prescription for high intensity statin therapy because we know that will help prevent the next heart attack by doing the intensive lipid lowering. The problem is adherence. A year later, a whole number of studies have shown that something like half of patients are no longer taking that statin. And so drugs don't work if you don't take them. Our thing would be to give them, before they leave a hospital, this one and done treatment that permanently reduces their cholesterol levels and takes that burden of adherence off the patients. They will be protected. And that would be transformative, I think. But then you're probably thinking, well, why are we waiting for heart attacks to happen in the first place? Why don't we stop them? Why don't we do primary prevention? I think we'll eventually get there. It'll probably be a decade or more before there's enough safety data to make people feel comfortable treated healthier patients. But eventually, I think we will get to the place as the technology gets better, as the economies of scale build, as the drug gets cheaper, as all sorts of innovations happen, because this is a rapidly moving field, right? We didn't really have CRISPR until just over 10 years ago, and now we're already treating people with CRISPR. And, who knows what's going to happen in the next decade. But I'm very confident that, 10, 20 years, we'll be in a position where we'll be able to offer a treatment like this to any young adult who wants it. It'll be up to them whether to take it. But if enough people take it, permanently reduce their cholesterol levels, push off heart attacks by decades, it would improve life expectancy. It would perhaps eliminate ischemic heart disease as the leading cause of death worldwide. It would be transformative for human society. So anyway, that is my story. And I'm happy to take any questions. I probably talked longer than I meant to, but
no problem. It was a very interesting story, and we do have some questions, and I have some prepared as well. Maybe, so there's some that's sort of on your career, and then some that are on the sort of promise of this editing approach for cardiovascular disease. So first, a question from our audience, or a couple questions from our audience. First, curious about some of the challenges you might have faced in sort of taking a technology that was sort of developed in academia and then needed to take that next step of going into a company, thinking about communications with the tech transfer office, those very early days where like this is just a sort of nascent idea and you need to build it into something that's a little more real. Could you speak a little bit about that process, particularly as a professor at the time when you were doing it?
Yeah, so I was sort of in an unusual situation. I had not actually filed any IP at Penn on this. What IP I had filed on the use of CRISPR was actually when I was still at Harvard, so it belonged to Harvard. The company, when we started it up, we needed access to Cas9, and that meant Broad Institute, Harvard, MIT, like jointly those institutions. And so we licensed it. We licensed Cas9 for use with PCSK9, with ANGPTL3. But since we ultimately ended up using base editing, we need more IP on top of the foundational Cas9 IP. And so the exclusive licensor of base editing technology, again from the Broad Institute, because it was developed by David Liu at the Broad, was Beam Therapeutics, a company that was founded a couple of years, or maybe a year and a half before our company was founded. So we approached them early. Once we decided base editing was going to be our thing, we approached them early, and we worked out a licensing deal. And it ended up being, without getting into the gory details, some royalties and also some opt-in rights they could pay for a third of the cost of development, and they would get half of the US profits if they opted in. Now, what's interesting is years later, actually just late last year, they ended up selling those opt-in rights to Eli Lilly for hundreds of millions of dollars. And so now Eli Lilly has the rights to opt in. And that's actually, from Verve's perspective, a much better partner because they have a very, very, very large coffers to draw from to fund cardiovascular outcomes trials, which is eventually what will be needed. And so that's how we did. And then for the LNP technology, we licensed that from a company called Verve Therapeutics. They're the same company from whom Pfizer and BioNTech licensed LNP technology for their COVID-19 mRNA vaccine. So there are actually some shared components between Verve 101 and the Pfizer vaccine, as it happens. So we were not about creating a platform. We were very product focused. We wanted to target PCSK9, ANGPTL3. We did the bake-offs. We decided what the best technologies were, and then we just in-licensed them.
Gotcha.
And then we were off to the races. And, some companies don't do that. They do their thing, make the drug, and hope they can evade the IP somehow or the other. And sometimes they are able to get away with that. Sometimes they are not. But we took a very above-the-board approach.
Gotcha. So a follow-up question, one that's a little more scientific, and then one that's a little more clinical. Something that I was personally curious as you were going through your talk, the mechanism of action on the ANGPTL3 knock. So it
Yeah.
seems like it's an editing approach that's more generally applicable, and you can make the argument that maybe in retrospect that would be the first thing to focus on if you had the ability. But is there anything known about how exactly it's sort of LDLR agnostic and how it's
Yeah.
working?
So it has multifactorial effects. One effect seems to be reducing VLDL particle secretion from the liver, so the production side. But it also does increase the clearance of LDL particles. It's just not through LDL receptors. It's not totally clear exactly how. It must be through alternative receptors.
Got it. Got it.
I worked on this in my academic lab for a while, and then it ended up being hard going, and then the pandemic hit, and that kind of disrupted
Right.
everything, and just never ended up really pursuing that further. It works, and that's really at the end of the day the most important thing. Right?
Exactly.
Yeah.
And I guess on the topic of things that work, I think for a lot of us who are clinical trainees or clinicians now, thinking about how to communicate these new sort of therapeutic modalities to patients and thinking about this is a sort of new concept, like
Yeah.
you're going to give me something, it's going to cause a permanent change. There's no going back. How do I sort of communicate this sort of new approach, whether they're a risk or not,
Yeah. I mean, you have to explain the benefits and risks, like, informed consent, particularly in the clinical trials, but even afterwards, it's going to be a therapy that's substantial, and you're going to have get informed consent, just like you would with any procedure. Actually, let me show a slide. There's already a lot of excitement about CRISPR, and patients are already starting to hear about it and ask about it and know about it, read about it in the news. I've read an article in Wall Street Journal just yesterday talking about Verve and gene editing. Interesting timing. Here's a poll that was taken just last year from patients about lipid-lowering therapies, offering them various options. And you can see there, once daily oral pill, and that'd be kind of like the equivalent of statins. A one-time gene or base editing treatment, that'd be something like Verve 101. Twice annual subcutaneous injections, that's kind of like inclisiran, the siRNA drug that's been approved for inhibition of PCSK9 and is available. Twice monthly subcutaneous injections, that's kind of like the monoclonal antibodies that inhibit PCSK9 that are already on the market. And what you can see here is there's no one modality that wins out. As a group, patients like choices and they have their own preferences. And what you see here is that the clear winners are, on the one hand, taking pills every day. A lot of patients are very comfortable with that. They already take lots of pills every day. What's the big deal about adding another one or two? And then there are a big group of patients, almost as big, who are really, as long as it's safe, would prefer that one and done modality. I think people, if you kind of, present it not as a standard medical treatment, but more like a procedure, almost like molecular surgery, almost like a vaccination. I think, well, maybe not a vaccination given the current climate today, but let's say molecular surgery. You get stents in, that's a permanent thing. You're making a permanent change. You get cabbage surgery, that's a permanent change. Patients accept that because they understand, hey, I'm doing something very important that's going to benefit me for the rest of my life. So I think if you communicate in those terms, you can get real traction.
That makes a lot of sense. And the survey data is very interesting. I haven't seen anything that sort of directly compares the editing to siRNA approach like this.
This wasn't us. This was like some other group that independently decided to do this. But hey, we'll take advantage of it.
Yeah. Another follow-up question, going back to some of your career journey discussion. You made this comment about being a serial
monotasker,
multitasker that I found very interesting. And I think a lot of us could benefit from hearing a little bit about how you've approached that. Like as a physician scientist, and for you specifically, you live such a varied career. And having the ability to do that serial sort of pursuit of your work versus resisting the urge to like sort of take on multiple things, try and keep multiple balls in the air. Can you speak a little bit more about how you balance that in your career?
Yeah, absolutely. So the best example is how I divide my clinical time up with my research time. So the vast majority of my time is research. There's just no getting around that. I spend a month a year on inpatient clinical service, and that's what I do. I do not do outpatient clinic. Inpatient service, 1 week, at most 2 week blocks at a time, so several times a year. When I'm on clinical service, I plan it very well and make sure it's during a time when, there's not major academic conferences I have to go to or this or that. Occasionally I have to take a holiday shift. That's okay. But when I'm on clinical service, I'm totally locked into being on clinical service. 100% of my energy and my time and my focus is on patient care. People in my lab, they know like I'm not going to be around. Like they might see me around since it's all at Penn and it's all like in the same building complex, but they know not to bother me. They know I'm not going to be meeting with them like I regularly do. I try to cancel as many meetings as I can from the ordinary. You can't cancel them all, but most of them. And like 90% of my attention is focused on patients and teaching the house staff and doing all the things that an academic cardiologist should be doing in that setting. And then when the 2 weeks or even 1 week is up and I'm off service, switch back full time to research. Now, I know a lot of physician scientists, try to do like the weekly clinic. Now, maybe they can make it work, but that would be devastating for me. And I think a lot of my success is because I have not ended up with that recurring distraction and having continuity care, being responsible for patients forever and being at their beck and call at any given time, right? I have chosen to regiment in a very, very strict way so that when I'm on, I'm on. When I'm off, I don't have to think about clinical medicine at all.
Okay, noted. I think that's advice that I'm taking my future clinical career. And then maybe a couple more questions to wrap us up.
Sure.
Drawing back to another comment you made about not necessarily needing to be intrinsically brilliant, but just being at the right place or being able to take advantage of the right opportunities at the right time. That's a theme that came up at multiple points in your career. And I think from someone in my perspective, or someone who's sort of at the trainee stage, looking at someone like yourself, it seems like, oh, this was like a magnificent career. Everything sort of worked out perfectly, sort of thing. And it's maybe to some degree intimidating to try and figure out, or like be in a position where it may just be that one thing or that one moment that sort of catalyzes things. So could you speak at all for someone who's in those sort of shoes, like so earlier in their career, trying to find that sort of catalytic moment, how to sort of approach keeping an open mind and like pursuing things rigorously?
Yeah, I mean, there are brilliant people. I'm just not necessarily one of them. In my case, I think it was more about, having the prepared mind, right? Like Chance favors the prepared mind, and being very open to new approaches, being a very, very early adopter. When I see the potential for something to have an impact, um, in a way that had not been possible before, I jump on it. And sometimes it works out, sometimes it doesn't. But I try, I go all in. I'm not standing back. Um, and I think the other aspect that has helped me is being cross-trained and not like, getting trained in just one discipline. But I think you advantage yourself enormously if, in my case, I learned human genetics, but I was also, getting my hands wet doing stem cell biology and keeping my hands in the wet lab. Um, and, keeping up those skills, picking up those skills in the first place, because that makes you unique, right? Because now you are the one who uniquely can draw from different disciplines and pull them together in service of answering a question or trying to solve a problem. Whereas otherwise you'd have to collaborate with other people. And my view is you are your own best collaborator if you can put yourself in that position. And by doing that, you put yourself in the best position to have the prepared mind if you do those things. And the last thing is, you have to look out for yourself and you have to be aggressive, right? And so the best example of that is my, basically bailing from Hopkins. It was on retrospect kind of a gutsy and slightly insane thing to do, but I took the risk and it paid off. And it wasn't that risky. I mean, I was trained and I would have gotten a job somewhere, probably in the MGH system if things had not worked out. But I was willing to take the risk, and it was a very well-reasoned risk. I put a lot of thought into it, but took the risk, and it ended up working out very nicely. So
Yeah. Yeah, and I guess on that topic of taking a risk and moving forward in your career, you mentioned at the beginning that you're maybe a third of the way through what you're doing now. In that last 2/3 or so, what are things that you're looking forward to doing and looking forward to accomplishing in the work that you're pursuing?
yeah, so I reached an interesting, juncture in the summer of 2021 when, the pandemic was sort of, not totally finishing, but, dying down and people were comfortable, returning back to the former way of things and my academic lab and stuff at Hopkins. I'm sorry, not at Hopkins, at Penn. Can you hear me? I feel like maybe I'm
We
cut
can
off.
hear
Okay.
you. Your
Okay.
video
The
has
video's
frozen.
okay, good. Sorry
So,
about that. As long as you can hear me. So my academic lab opened up at Penn again. And it was really this nice opportunity to reboot. I lost some people through.
sorry, everybody. I think he'll be right back. But I guess while we're waiting for him to pop back up, thank you everyone for attending. We'll send out a short survey about the session just to get your feedback about it, as well as information about some of our future events. And hopefully he'll come back to share those last couple comments. But if he does it and you happen to jump off, thank you very much for attending.
So sorry about that.
No
My
problem.
Wi-Fi died, so now I'm talking to you on my phone. But I do want to answer that last question for anyone who's
Yeah.
still on.
Yes, please. Yeah.
So academic lab opened up again. All the restrictions were lifted at UPenn, and I'd lost some people from my lab through attrition. There was a hiring freeze, so I couldn't hire any new people. So it was this very unusual opportunity after a year and a half away, physically at least, from the lab, to actually reboot and decide, hey, I can work on something entirely new if I want to. You don't get that opportunity very often. And I was at this interesting juncture, as I was saying, where I'd kind of completed the full cycle from discovery all the way to, actually getting a drug into the clinic, or at least we're on the path to doing that. And so my role in that was sort of done. So it's like, okay, what's the next challenge? And what I realized is that from my time at Verve, I'd learned how to do drug development, and in particular, how to do editing in the liver. And so the next challenge I decided to take on is asking, let's think broadly. I don't have to restrict myself to heart disease. If I can correct any disease by doing editing in the liver, let's go after it. And so this has gotten me interested in inborn errors of metabolism, diseases like PKU, like urea cycle disorders, organic acidemias, and a whole host of disorders, where if you can make a corrective edit for a pathogenic variant in the liver, it would have a substantial therapeutic effect, potentially be curative, and address enormous unmet medical needs. So that is largely what I'm working on these days. It's not all that different technologically from what I was doing, what I learned how to do at Verve and can now actually do in the academic setting. Before, I would not have been able to try to develop drugs in the academic setting, but now I'm starting to think I maybe can. And what is really facilitating the process is my being able to get very, very large grants from the NIH to support this work. So, for example, last year got a $26.5 million grant to take, one of the PKU therapies into clinical trials. And so that's what's going to occupy my attention over the next few years.
Yeah. And hopefully there'll be more curative drugs to come out of that.
There will be, 100% sure of that.
Yeah. All right, Dr. Musunuru, very thankful for your time here, and I think our attendees as well as myself really gained a lot from this. And yeah, thank you so much for lending your insights.
Yeah, thanks so much for having me, and my apologies again for the little glitches, but I think
yeah, I think we made it work. All right, and thank you so much to everyone for attending. Stay tuned for information about future sessions, and we hope to see you there. All right, thank you.